B, and C. So these three people are committing a crime. Okay, these three people are involved in a crime, not committing a crime. So who is that individual? So that individual who has been killed is called as C. Okay, the C is dead. So when C is dead, so A has killed C. So that was our story. So now what happened? C's body is found in somewhere in the deep forest. Okay. So in the deep forest, his body is found, but unfortunately, it's been certain days, several days since his body was discovered. Okay. So what has happened? They've killed him. So the coyotes or the foxes or whatever it is, they have taken his body and eaten his flesh. Obvious results. So now, because of the smell, the police is called and somebody spotted the body, and you come to the location. Now, how do we investigate this crime site? So first thing that we do is profiling. So to do the profiling, we use various techniques in the forensic department. So what you're learning is criminal forensics or forensic investigation or crime forensics. So where you're investigating a murder, okay, or anything. See, if the victim is a female, then we look at two situations. One is whether it is a murder and rape or it is just a murder. If it is male, we'll look at murder, okay? So here, let's say that the victim is found. Now, what happened? Let us take a female as an example because it will solve two situations at a time. So now what we will do is, around the perimeter of wherever this person is being killed, we try to understand how they entered the situation, how they entered into the crime situation. When we try to build the story of how they got into a fight and who attacked who and where the fight started and how they started fighting. So you construct a story. Now the nearby inanimate objects like the rock or the knife or the stick, or there is a cloth that is stuck to the tree or a branch, okay, or sometimes when they are pushing and growling, a tissue is rubbed against the concrete, or a tissue is rubbed against the bark of the tree. You understand my point? So when somebody is trying to kill you, would you just stand quiet? No. There is high chances that you hold this person. Maybe there is some flesh left in the nails. Okay, there is maybe there is while fighting, somebody grab the hair and they pull the hair, right? So now what happened? Every single a small tissue can solve this entire problem. So there is a story. I went to this forensic lab in Bangalore, so which deals with crime, criminal investigation. So my question was I was very curious, how to solve a crime. So I asked the person who is a forensic investigator, how do you escape a crime? So he looked at me and he says, there is one way to escape a crime. That is, if you can able to burn a body. Okay, I'm not teaching you how to commit a crime, but I'm telling you a situation. Okay, if you're able to burn a body, whoever has been killed, up to 98% or 90% or above, only bones and not even bones are left. Okay, if you're able to burn him like that, then you escape a crime. That is high probability, because you won't get any samples. DNA tissues is very hard to find, and if you should have like cleansed the entire crime scene. So that is high possibility. That is, let's say that there is 70-30. 70% you won't be caught, 30% there are high chances because of your stupidity or because of your anxiety, or you're going to get caught. Now, how to escape a crime? It's a very difficult question. Okay, but this is a high probable scenario. Other scenario is, second question I asked is, let us say that I committed a crime and I try to wipe the entire region very cleanly. I'm very thorough. Okay, and I clean the entire crime site without leaving a single tissue. I have not left a single tissue there. Okay, how do you catch me? So that means I'm asking him indirectly, how much quantity of DNA do you need to catch a criminal? Now the expected answer is very, very shocking. Even that made me leave hopes on committing a crime. So what he said was, you need only 53 picograms. Okay, only 53 picograms of DNA to commit a crime. People, 53 picograms is less than a nano. So if it is 53 picograms, imagine, like while I breathe, I think that I have exchanged some micrograms of DNA. Now 53 picogram is so negligible. You can't even see, you can't even feel that you have left it. So your chances of getting caught is 100%. So how do we catch is the big question. So first one, what do we do is we do swabbing. So this process of learning how to collect, okay, how to collect the samples from a site where you have committed crime is called as swabbing. So what is swabbing? How do we do swabbing? Swabbing is very simple. I mean, like, it's complex and simple at the same time. You take the swabs, like cotton swabs, and you try to, for example, I told you, like, let us imagine that a female is being murdered, so that it is all two situations at a time. If the rape and murder is being committed, now what will happen? The tissue will be exchanged within the individuals, like vaginal stabs or swabs will be taken, okay, then semen samples will be taken, or the samples that are stuck in the nail will be taken, okay, and everything, okay. If it is a murder, the knife or the blood samples, wherever there is a spillage of blood, the samples will be taken from that. If there is any tissue stuck, or if they have rubbed their bodies against anything, there the swabbing will be done, okay. If there is a finger mark, or if they have like tried to put their hands to take grip or anything, there the swabbing will be taken. So several places we collect the swabs. So once the swab is collected, we definitely, there is 100 highly high probability that at least in one of them we have collected the sample of a criminal who have committed the crime. So now what happened is, now we try to sample the DNA. So how do we sample the DNA? First one, we have all our DNA within the cell, correct? So it is in, let us assume that it is within the cell. So if it is within the cell, then we have to do something called as isolation. So how do we isolate DNA? DNA isolation can be done by enzymatic digestion, right? So we do enzymatic digestion. How do we perform enzymatic digestion? Your cell is made up of biochemicals. We know that, whether it is carbohydrates, lipids, okay, or it is proteins, whether it is any type of nucleotides such as RNAs and DNAs. So we try to enzymatically digest the cell using all the different types of enzymes, okay. For example, proteases, amylases, carboxypeptidases, or we also uses RNases, except for DNA. Except for DNase, we use all the other enzymatic enzymes to digest the cell. So now in the suspension, in the cell meat, in the suspension, you will have a, you will have a test tube, and the test tube will be like in the liquid form. You cannot see basically. Now what do we do is we pour chilled ethanol. So when we pour chilled ethanol, okay, so when we pour chilled ethanol, now what happens is this will dehydrate our DNA, so that our DNA will start coming like a small mucus like structure, and it starts floating on the surface of your test tube, okay. It will come up. Now what we can do is you can just spool. Spooling is just, you can just twist and turn, and the entire DNA is stuck to that spool like a glass bar, and then you can transfer it. Now we know that our DNA is so large, right? Our DNA is made up of around 6 billion base pairs, like in a double stranded situation. Now what we have to do, we have to make sure that our DNA is put in, made into small pieces. So for that, what do we do is we are using restriction enzymes, okay. We are using restriction enzyme. So, and using the restriction enzyme or molecular scissors, okay. So when we use molecular scissors or restriction enzymes, now the long DNA that we have is cut into small fragments. So this is also called as fragmentation, okay. Now we have the fragmentations or fragments of DNA, okay. We have the fragments. Now just imagine, people, okay, we have the fragments of DNA. What all fragments, like all this process, like cell isolation, enzymatic digestion, and the DNA, you have it, and then you fragment the DNA. For what DNA you are doing is a very important question, isn't it? First thing, we have the dead person's DNA, and second thing, we have the criminal's DNA, okay, from from the crime site. So that is another thing. Next one, what do we have is we have a suspect's DNA. Okay, so who are our suspect who have committed the crime? We think that they have committed the crime using so many different types of investigations. So let us say that we have A and B have committed the crime. So these are our suspects. So we have their DNA. Now, what is our motto? We need to match the criminal's DNA from the crime site with the suspect DNA. And why did we need the dead person's DNA? We need to differentiate, isn't it? Whose DNA is whose. So once we have the sample that matches with the criminal, criminal's, dead person's DNA, so we eliminate this. We don't want this anymore. So we just want to match this criminal's DNA from the crime site and the suspect's DNA, and we subject both of their DNA into these processes. Understood? Now, once we have the fragmentation, these fragments have to be separated. Now, how do we separate this fragmentation? We have a process called as agarose gel electrophoresis. So what is agarose gel electrophoresis? Now we try to understand the physical properties of DNA and we use it against it so that we are able to use this process to separate DNA based on the size. So agarose gel electrophoresis is also called as size expulsion. Okay, size expulsion separation. Okay, based on the size we separate the DNA. So how do we do it? We first introduce our DNA. Let's study the first steps. Our first step is we take a box. Okay, a box. So basically it's not a box, we have something called molds. So these molds are just like how you create toys, sugar toys for your festivals. Okay, in Kannada we call it as achu. Okay, in Telugu we call it as pacha. I don't know in Hindi, I'm very poor at Hindi. So we have a mold, like brick molds, you see. So for that, we use something called as agar-agar. So this agar-agar or agarose, what we do is, we use this agarose and then we put it in the distilled water and we make a solution of it. So once a boiled solution of this agarose is there, it is in the liquid form. Now, slowly we pour this into the mold. Now, we pour it so carefully so that no bubbles are formed, and this will fill this mold. Now, we could have used anything. We could have used anything for the filtering. Why are we using agarose only? Now, if you look at the molecular structure of agarose, the molecules of agarose are so well organized. Okay, they're very evenly organized in their in their structure, especially if you study the solid state. You see how the atoms are arranged very systematically and symmetrically. So that is how the agarose molecules are arranged, and the arrangement gives a very, very specific intermolecular space. So the space is very, very even throughout this membrane. Okay, throughout this membrane from here to here or here to here, this size is very, very uniform. Now, having this uniform, now what will happen is it will allow the fragments to migrate based on its size. Understood? So that is why we use agarose. Now, once we have agarose, now we have a bed, we have a gel bed like this. Okay. Now we have a gel bed like this. Now, what are we supposed to do? We drill wells. Wells means make small holes. Okay, at one end of the agarose bed. And now I introduce my DNA fragments which are labeled. So I label this with some fluorescent dyes, let's say, because I should be able to see them, or radioactive dyes. Mostly we use fluorescent dyes. So we put fluorescent dye and we label our fragments and then we mix it. And then now what we'll do, we introduce our fragments into these wells and we fill them up. Now, how are we introducing this thing? Let's say I have A's DNA, I have B DNA in this well, and I have the DNA from the crime site. Understood? So I have the DNA from the crime site. Now what happens is, or I can neglect this crime site DNA also. Understood? Okay. So let's say that I have put A DNA and B DNA, and I have these two DNA I have. Now what I will do is I will try to separate them. Before I separate them, I have to put the electric charge. Now to put the electric charge, now if I put this in the solid state, it will burn. Okay, everything will burn. So what I will do is I will try to fill this up with a buffer. Okay, we call it as a buffer. So buffer will make sure that the electromagnetic field is evenly distributed. So the tank will be having the gel, and above the gel there are wells which are filled with our DNA fragments, and over that is my buffer that is floating. Now, what charge is your DNA? Negative charge. So we add it to the negative terminal. Our well is at the negative terminal, and this is the positive terminal. If I pass a specific voltage, okay, it's quite a high voltage. When I pass a specific voltage to my gel medium, especially, now what happens is at a certain voltage, my fragments will start migrating towards the positive end. Now, once they start migrating towards this positive end, they give me beautiful bands. Okay, they give me beautiful bands. How do I see these beautiful bands? See, after 10 minutes or 15 minutes, I see that I feel that my agrogel electrophoresis is done. So what do I do is, once this process is done, I switch off the electricity. I switch off this electricity. Now, what do? I expose this to UV light. When I expose this to UV light, now all the fragments will start glowing in orange color. So once they start glowing in the orange color, now I can see that there are bands of these fragments which are very beautifully separated. Understood? Now, once they are separated, now I know that, okay, I have successfully separated this band based on the size expulsion. Now, what is my next step? Once I'm done with this gel, now I have to make sure that this gel is transferred onto a membrane where I can do my hybridization. So how do I do that? I will take my gel and I will place it on a nitrocellulose membrane. So once I place it on the nitrocellulose membrane, now what happens is my gel is very, very wet. Now, once I place it on the nitrocellulose membrane, now, a systemic weight is being transferred over a period of time. For example, it might be 12 hours or it might be 24 hours, depending upon the membrane or the gel we are using. So now we try to blot it with lots of blotting papers so that the water is removed, and now the gel becomes smaller and smaller and smaller as the water is being removed slowly. Now, once all the water goes up, your gel becomes such a like a paper thin, and now these fragments which are in the gel gets transferred onto a nitrocellulose membrane, and this process is called as Southern blotting. Okay. So in simple terms, we just transfer the fragments in the gel bar into the nitrocellulose membrane, and this process is called as Southern blotting. So for example, we have Northern blotting and Western blotting also. Northern blotting is for RNA and Western blotting is for protein. So that is not necessary for us. So now we have a Southern blot. And in my blot, in my nitrocellulose membrane, so I will try to erase this. Okay. Hold on. Yeah. Now, once I have my nitrocellulose membrane with my fragments, here are my fragments. Okay. Now, what do is I will hybridize them. Okay. Sorry. Now, how do I hybridize? I take the fragments of the DNA from the crime site and I introduce to this. When I introduce to these, now these are called as probes. Okay, so these probe DNA are from the crime site, so they will get hybridized to the one that are from the crime site. So A committed the crime and B did not commit the crime. Now what will happen is these probes start hybridizing with the ones which are exact match. Okay, now once this hybridization is done, I take an X-ray. So once I take an X-ray, now I see an X-ray and I can easily tell that if I conduct a murder, okay, if I conduct a murder, then obviously 100% of the hybridization match. Okay, 100% of the hybridization match. If I have maternal or paternal issue, that is, if the mother is claiming he's the father, okay, and the father doesn't know if he's a father or not, then you do the DNA test. Even if there is 48 to 50% match in child's DNA, okay, in child DNA, then sorry, in DNA, he's the father. You understand my point? If it is rape, again, it is 100%. So this is how you catch a criminal. And how much is the certainty of giving justice? 100%. There is maximum 100% times that this will never go wrong. There is one small instance where this can go wrong, where if a monozygotic twins, okay, if you have a monozygotic twin, okay, they have exact genetic makeup, you see. So when a monozygotic twin, if one of the twin commits the crime and the other twin is very innocent, now both of the DNA will match, and identifying the criminal is very, very difficult, in especially in monozygotic twins. Why? Because even their physical fingerprints are matching, understood, and their behavior is also similar. Who commits the crime is matters, not the behavior. So that time you should go for a lot of other profiling, okay, and it is very fascinating field of science, forensic science. So this is what is DNA fingerprinting. So we understood what is how to isolate DNA, how to fragment the DNA using restriction enzyme, and how to do conduct agarose gel electrophoresis, what is southern blotting, what is hybridization, X-ray, and coat, and the decision that comes. Okay, so this is about DNA fingerprinting. Did you guys understand this? Yep. Can we go for the next one? Yes. Thank you, people, for the response. Perfect. Okay. I wish we had more time, because it's nice to discuss some cases, right? So we could have taken like few cases and discussed, okay, this happened in this case. I have like so many good examples, with especially with respect to case studies in forensics, and it's very fascinating. You will be really surprised how on earth even people think to commit a crime like this. It's very nice. Maybe sometimes after your exams, when we come back, we will discuss about the forensic. If you're interested, we will discuss about it. All right. So for us right now, I'm going to conduct a very quick crash course on this lesson called as Principles of Biotechnology, or Biotechnology Principles and Processes. So let's go. Hold on. Here we have. Let me unblank the screen. Chalo. So I think you might have heard about what is biotechnology and what is this principles and processes. Now, when it comes to biotechnology, it's not today we have been using biotechnology. I can tell that we have been using this from thousands of years, but we call them traditional biotechnological tools. And here today, modern biotechnological tools is very, very scary and also very fascinating at the same time. So back in the days, when you see that our ancestors, I don't know about you kids, but we could see in our time, we would take these bulls and cows and sheep to a specific village in other district, and we would be having this fair. Fair in the sense, like every village, other villages from different villages, they would gather there, and we would specifically look at the bulls and the cows and the sheep or the goat, and we would look at their genetic characteristics, and we would ask how much milk does it give, what is the age, and look at his muscles, and we would say, oh, this is the guy, he is very dominant and we need him, or this lady or the cow, she is having a high potentiality to give more milk, maybe she will give better kids for us. So that is how we used to choose the breeds, and then we used to cross, whether we would do cross breeding or interbreeding within the species. So we would do this to get the high yielding variety, right? And sometimes my mom used to teach me how to do this grafting using the rose plant. And we also used to try to graft with hibiscus. We used to be so excited if we get the red rose and the pink rose and the white rose in the same plant. We would think that we are genius, exceptionally brainy people. So that's how our childhood was. And now we realize that all these things were like primitive biotechnological tools that we used maybe in the past, or even in the villages that we use even today. But the modern biotechnology, it involves the live organisms or the enzymes of the organisms, and we alter them for the human use. Okay. So the European Federation of Biotechnology defines biotechnology as it's an integration between the natural resources, such as organisms, cells, and their parts. Uh, we use a molecular analogs for their products and services. So this is called as biotechnology. So definition might be different. I mean, like definition might be hundreds, but the principle behind it is very, very similar. So what are we doing? We are using the technology and we use the organisms for human benefits. It's as simple as that. Whether you produce insulin, whether you produce toxins, whether you produce antivenoms, whether antibiotics or vaccines or anything you talk about, it's all about the applications to for the human benefit. Not none other than that. So today we use biotechnology for so many different fields. For example, we call it as medical biotechnology, where we use for diagnosis. For example, in COVID, we try to diagnose using kits, right? So rapid kits, rapid antigen testing kit. So that was developed during the COVID time. Is nothing but biotechnological tool where we produce some proteins which could recognize this virus is present or not. Okay. Similarly, we used RT-PCR techniques. So that is also biotechnology, right? So then we produce vaccines. That is also medical biotechnology. So next, animal breeding and agriculture. So today, we have such efficiency to even look at a single genome that is present in an organism and try to build a proper analogy on what this organism is really is, and whether it is a good breed or a bad breed, and what are its characteristics at a genetic level. So we use this for animal breeding and to preserve animals also. So today we are doing so much of struggle to preserve our native breeds with respect to cows and buffaloes, as well as sheep and goats. And we are also trying to bring certain country like Afghan goats into the Indian soil because they have good meat production in a very short period of time, something like that. So there also we are using biotechnology for animal breeding as well as in agriculture. Healthcare and pharmacy, that's what we talked about. Improvement in food technology. For example, we are trying to today, when the rice we eat is not just made up of carbohydrates, it is made up of many other things. For example, proteins, vitamins, and minerals. And why are we getting all these things? We are not supposed to get it naturally, but we are getting different types of these sources of minerals or good nutrients because we are trying to fortify. We call it as biofortification. So this biofortification or putting more minerals content or more nutrient content within a single crop that is affordable for the human, so make sure that every single human being is nutritious and they are not malnourished. So that there also we are trying to improve the food quality. The milk we drink is also biofortified. Okay, the crops we are eating like rice, ragi, and millets, they are also biofortified. So etc. Next one is fermentation technology, which has been highly used in the production of wines or any brewery, or for example, we also use it for the production of certain antibiotics like penicillin, etc. Next one, recombinant DNA technology, where we try to do genetic engineering to create recombinants for so many different purposes. Especially they are used in medical biotechnology for the production of drugs, vaccines, etc. Next one is plant agriculture and crop improvements. We already studied about it a little bit. Next one is environment and energy management. So environment is we are trying to create some fungals, or not create, we are trying to enhance certain fungal species so that they have the capacity to digest this oil spills that are done in the sea, or the ones which have the capacity to digest the plastic that is discarded. So that is also being used. And energy management, like some of the algae, we're trying to do bioengineering so that they are capable enough to produce biofuels so that we reduce our dependency on fossil fuels. Okay, so that is also happening. Chalo. So yeah, this one, yeah. Next one. What do we do in biotechnology? Basically, biotechnology involves genetically modification of organisms or GMOs, creating a GMO. So here, first we identify the gene of interest, or for example, what do you call as a gene of interest? Let us take an example. If I have a huge population of human beings that are suffering from a particular congenital disease, such as diabetes. Diabetes is a disease, right? Why do people suffer from diabetes? Because they don't have the capacity to produce a healthy insulin. Now, how do we produce insulin? Insulin is nothing but a hormone, and it is made up of proteins, and it has a gene which is present in the human being. Now, can insulin gene be a gene of interest? Yes, absolutely. So we can use this gene of interest, which is an insulin gene, and can we find the methods to produce artificial insulin or insulin outside human body? It is possible. Now, insulin becomes the gene of interest, and human beings becomes the organism of interest. Okay, one second. Yeah, sorry, people. Let's get back. Organism of interest. Now, once I have the insulin, healthy insulin gene, what can I do with it? Now, let us say that I think that I can produce an insulin using a bacteria. Is it possible? 100% possible. Now, how do I do it? Now, the first thing that I have to do is I have to make sure that I have the technology to introduce my human insulin gene into a bacterial cell. Now, once I have the gene into my bacterial cell, that is not the end of the show. That is the beginning of the show. Once I have put my gene of interest, which is a foreign gene, into a different organism itself, now I have to make sure whether this organism survives. First thing. Second thing, can my organism, who has the gene of a human being, produce certain proteins that are of my interest? Can the gene be maintained without being harmed? And can this organism, which is carrying my foreign gene, make multiple copies of itself? I cannot keep on editing every single bacteria, right? So what I can do is I can use the advantage of asexual reproduction, where this bacteria goes into binary fission and produce clones. What are clones? Genetically identified organisms are called as clones. Now, if I have my gene of interest, definitely these clones should be able to make the copies of itself. So that is very, very important, whether it has my gene of interest, whether it can produce a desired product, and next, whether it can multiply itself into, copies or make generations of itself. So these three are important aspects of creating a genetically modified organism. After that, we have several steps and we'll discuss further about it. Now, once we have these three things, okay, then we are good to go. So for the first genetically or recombinant DNA technology was done by Stanley Cohen and Herbert Boyer. So they produced this first recombinant DNA, and for that recombinant DNA, they used this organism called as Salmonella typhimurium, and they transferred this Salmonella typhimurium's antibiotic resistant gene into an E. coli and made it antibiotic resistant. So we'll discuss about it, no problem. So what are the steps or what are the tools that are required for creating a GMO or genetically modified organism or a recombinant DNA? So for that, we require three major tools. One is a restriction enzyme, second one is a cloning vector, and third one is a competent host. So let us discuss one by one. When you talk about restriction enzyme, where are this restriction enzyme coming from? So basically, majority of the living organisms, especially the ones which are unicellular, they have restriction enzyme inbuilt. Naturally, they have restriction enzymes. Why do they have restriction enzyme? And what are restriction enzymes? See, these are specialized enzymes. They are meant as a cellular defense. So restriction enzyme are present as cell defense in organisms like bacteria and protista and fungi, etc. Okay. So what do they do? Why do they need this? So these restriction enzymes, they have the capacity to identify a specific sequence of the DNA, and then they make sure that they cut this DNA on the recognized sequence. So if it is a sequence of the DNA, and if they recognize sequences like this, so they have the capacity to cut that specific spots. Now, why would somebody keep an enzyme that can destroy some DNA, right? Cutting the DNA means destroying the DNA. So they have this particular mechanism because these unicellular organisms are a huge target for viruses. So virus, what it does is, the virus has a capacity to survive by injecting its foreign DNA. Remember? So the viral DNA should be destroyed before it gets into the host DNA. That is why what happens is, these organisms who have the restriction enzyme, they produce this restriction enzyme to recognize sequences in the viral DNA, so that they cut this viral DNA and destroy it before it takes control of the cell. So that is why I mentioned that in the beginning itself, that it is a part of a cellular defense when it comes to the restriction enzymes. Now, usually what kind of sequences do they recognize? Now, this restriction enzyme, they are having the capacity to recognize a specific sequence called as palindromic sequences. Now, what is a palindromic sequences? Palindromic sequences is something that means the same when it is read from 5' to 3' direction or 3' to 5' direction, and we'll discuss about what are those sequences. Understood? So, what are the types of restriction enzyme we have? We have three types of restriction enzyme: type 1, type 2, type 3 restriction enzyme. So in restriction enzyme, the major category of restriction enzyme is of two types. One is called as exonuclease and other one is called as endonuclease. The exonuclease version of the restriction enzyme, what it does is, let us assume that this is our gene. Okay, this is the gene, and our exonuclease has the capacity only to cut at the ends. You see, it only cut at the ends. That is its only capacity. So that is why it has very less application in genetic engineering. So we don't use it much. Whereas when it comes to endonuclease, right, endonuclease, so our endonuclease restriction enzyme, they have the capacity to identify the sequences between the genes itself, and they will be able to cut in the center. Okay, in the center part of the gene, they are able to cut. So this application gives us a vast amount of scope to edit something, right? So that is why we use endonucleases. In endonucleases, we have three types: type 1, type 2, type 3 endonucleases. The major type of endonuclease that we use for our genetic engineering is type 2 restriction enzyme. Remember this. Type 2 endonucleases is used at the maximum extent. The reason is, it is highly accurate and it is highly efficient in identifying the sequences and in cleaving the sequences to our requirement. And what is type 1 and type 3? Right now, it's not necessary. When we have time, we will discuss in detail. Understood? So now we know that restriction enzymes are of two types: exonuclease and endonuclease. And endonuclease are the one which cleave in the center or cut in the center. So they recognize palindromic sequences, and we have three types of endonucleases. Among three types, we have using type 2 type of restriction enzyme. Understood? So once we have a restriction enzyme, I told you already, restriction enzymes are not man-made. Restriction enzymes are isolated from a foreign source. So what is a foreign source? Our foreign source is a bacterial cell. Our foreign source is a protestant cell. So wherever we are isolating this from, so that is our source. So once we isolate the restriction enzyme, today we have around 600 different types of restriction enzyme, and each of the restriction enzyme has a specific capacity to identify a specific sequence and to cut the specific sequence. Understood? So how do we differentiate between one restriction enzyme to other restriction enzyme? So to do that, initially, we already know the characteristics of an enzyme. Based on the characteristics of an enzyme, excuse me. Sorry, students. Okay. Based on the characteristics of an enzyme, we don't name them, but based on few things, we try to label them. What is that? For example, we have a restriction enzyme called as EcoR1. So EcoR1 is a type of restriction enzyme. So how do we name this? E is for Escherichia, which is a genus name of a bacteria. CO is coli, which is a species name of the bacteria. R is the strain of a bacteria, and 1 is the serial number of which this enzyme was extracted. EcoR2, EcoR3 means EcoR1 came in the beginning and followed by 2 and 3, etc. Understood? So this is how the naming is done. So for example, you see E. coli RY30. So that means ECO R13. So that means we are talking about a specific type of a restriction enzyme which is isolated by E. coli. Understood. Similarly, we have HIN3, BAMH1, SAL1. So these are different types of restriction enzyme which are isolated from a particular source, and this is how we name it. Understood? So coming to the action of restriction enzyme, I told you already, guys, that restriction enzyme can identify a palindromic sequence. Correct? So in your book, they have given a palindromic sequence as Malayalam. Correct, right? So it says that if you read from this direction also it's the same, or if you read from this direction also it's the same spelling. So this is what we call it as palindromic sequences. But sadly, my students will get confused, and I don't like to confuse them. If you look at the recognition sequences of a specific restriction enzyme, for example, E. coli or HIN3 or BAMH1 or HA3, so look at the sequences. If one strand is going from 5 prime to 3 prime end, and let's say that one strand reads GAATTC, the opposite strand in the opposite direction will read the same, right? So that is what? GAATTC. So this sequence is called as a palindromic sequence. You understand? Now what we think, okay, AGCT, TCGA, it doesn't make sense, isn't it? So that is why we have to consider both the strands. So if we consider both the strands, then we get a proper palindromic sequence. So AATTC, and then you see AATTC. So this is called as a palindromic sequences. So when they recognize a specific palindromic sequence, see, I kept mentioning that it is specific palindromic sequence. See, E. coli, ECO R1 can only recognize this sequence such as AATTC, and HIN3 can only recognize AGCTT or AGCT. Next one, BAMH1 can only recognize GATC. Okay, something like, they are very specific. They do not even accept a single change. Even if there is a single change, these sequences do not get recognized and they do not get cleaved or cut. Okay, now how do they cut? Now, if you look at the arrow marks that are given in this picture, what you can see is, you can see that some of this restriction enzyme, correct, such as ALUL. So how are the ALU1, sorry, how are they cutting? You can see that they are cutting exactly at the same site, or even H3 is cutting at the same site. Whereas BAMH1, it is cleaving at two different sites. So this gives us two different types of sequences. Now, if a sequence is like this, and if it is cut at the same point, these are called as blunt ends. These are called as blunt ends. And if you have a sequence like this, and if it is cut like this, now I would get a sequence something like this. Correct, right? So I would get something like this. So this is called as sticky ends. Or here, yeah, this is called as sticky ends. So these are called as sticky ends. Usually what happens is, while I am doing the genetic engineering, if I use two different types of restriction enzyme, okay, imagine one restriction enzyme goes for blunt ends and another restriction enzyme goes for sticky ends. Can I stick this together like a paper? Okay, imagine this like a paper. Okay, so let me erase this. Okay, this is like a paper. Imagine, can I stick this together? Is it possible? No, this would be left out. Now my sticking together plan, the plan of sticking two ends together will not work. So that is why during genetic engineering, I would really look into what type of ends my restriction enzyme create, whether it is sticky ends or it is blunt ends. If it is creating sticky ends, I would go for the restriction enzyme that will give me more sticky ends, so that I can easily put my DNA, which is of this shape rather than blunt shape. Correct, right? So it is easy for me to stick a DNA something like this. So this will patch. So now my two fragments will stick together. So that is why when I'm doing my genetic engineering, what will I do? I will go for more of sticky ends. Did you understand why sticky ends and why blunt ends, and what are palindromic sequences and what are restriction enzymes, right? Unfortunately, we ran out of time. Time runs so fast here. All right, students. I have to conclude the class, and we will meet in the next class. Okay. Probably after your exams. All the best for your exams, and study well, do well, don't take tension. Calmly you finish your examination, and we shall meet after that. Okay. All the best. Have a nice weekend. Bye bye. Take care. You're welcome, people.